Assalamualaikum and hi, I'm Najihah from AS2016A. In this video, I would like to present about my research, which is effect of different nitrogen concentrations on the morphological growth of eggplant, Solanum melongena. Eggplant or brinjal with its scientific name Solanum melongena is one of the agricultural products that have a high commercial value, especially in Asia. Based on statistics, eggplant is the fourth of demanded vegetable in Malaysia because the production is about 41,157 tons in year 2019. In 2030, world population is expected to increase to 8.6 billion, so demand for raw materials such as agro-based will increase too. Generally, in this study, the growth development of Solanum melongena were analysed under different amount of nitrogen. So nitrogen was chosen in this study because it is one of the important macronutrient needed by plant, which contributes about 1.5% to the plant. Unfortunately, there is a need for measures towards the nitrogen level applied in eggplant crops. It is important to know the optimum amount of nitrogen concentration needed by the eggplant as well as to study its vegetative growth. The main goals for this study are: first, to investigate the effect of different nitrogen concentration on the growth rate of eggplant seedling. Second is to determine the effect of different nitrogen concentration on the root and shoot biomass. And the third, the last one, to determine the effect of different nitrogen concentrations on the vegetative and flowering stage of eggplant seedling. Eggplant, or the scientific name is Solanum melongena L. It is also known as brinjal or aubergine and is a nightshade plant from the Solanaceae family. Eggplant diversity is primarily to several countries in Southeast Asia, includes India, China and Indonesia. The fruit of this plant are varying in colours from black, purple, green, yellow and white, while the shape is oval shaped to long shape. Eggplant is kind of vegetable that good for human health because it contains low calories and high mineral sources. The growth phase of Solanum melongena plant consists of 4 stages. Firstly is seedling stage, vegetative growth stage, flowering stage and harvesting stage. If the eggplant successfully produces fruit buds, it indicates that the growth is at the highest. The other main thing is nitrogen element is the key nutrient in plant physiology. According to Sashi 2021, nitrogen function in enhanced photosynthesis, increases flower productions and increases crop yield. Nitrogen helps to synthesize amino acids to form proteins, which is highly demanded by plant. Nitrogen play an important role to the plant since the growth and production of eggplant depend on protein stored. And next, nitrogen fertilizer is divided into 2 types, namely inorganic and organic fertilizer. The process of inorganic nitrogen absorption is called oxidation. Besides, excessive application of nitrogen fertilizer on plants can cause them to accumulate in the soil and then causing environmental problems. Thus, the way to prevent the absorption of excessive nitrogen components by plants is with efficient nitrogen management. And the last one is increase in nitrogen supply can increase the plant characteristics such as plant height, number of leaf, leaf area, stem diameter and many other characteristics. It indicates that plant growth responds well with the higher level of nitrogen applied. However, higher nitrogen application can also give negative impact on plants. It will reduce the physiological and chemical properties of plant such as chlorophyll content and seed numbers. Lack of nitrogen can make stunted plant growth and slow life cycle phase, which then causing decreased amount of crop yield. Next, I will continue to the methodology parts. First thing first is material needed, of course, Solanum melongena plant. So 15 healthy eggplant seedlings with the age of 30 days were obtained from the nursery. Okay, the experiment was conducted at Laman Agrotech UiTM Jengke Pahang for 5 weeks and the plant is arranged as in the picture. Eggplant seedlings were transplanted in 16 bud multiply 16-inch polybags which contain sandy clay loam soil. Eggplant seedling was watered twice a day with tap water, and agronomic practice such as pest and disease control have been performed. So in this study, urea fertilizer was chosen as nitrogen source. So 4 different amount of urea fertilizer were used as treatment, which are 3 mg as treatment 1, 9 mg as treatment 2, and 50 mg as treatment 3. And another one is treatment 0, which is 0 mg as a control treatment. These treatments were applied on Solanum melongena every 5 weeks of treatments. And then we move to data collection for few parameters studied. So the first one is plant height. Plant height was measured using measuring tape in centimeter scale from above ground to shoot parts. For stem diameter, it was measured by digital vernier caliper with millimeter units, and the leaf number was counted directly for each eggplant. All these 3 parameters were analyzed their relative growth rate (RGR) by using this formula. Okay, next, for plant dry biomass, the plants were cut into 2 parts, above ground and below ground, and then washed to remove soil residue. The plants were dried using an oven at 65 degrees Celsius for 3 days. And the last one is the vegetative and flowering stage observations. The appearance of the buds and flowers have been observed when the plant reached at flowering stage. All the collected data was recorded in Microsoft Excel and analyzed using one-way ANOVA by using SPSS software with 5% significant level. Okay, firstly, we look at the growth pattern. For plant height, 0 mg of urea is the lower height growth trend for Solanum melongena. The trend illustrated that 9 mg of urea produced the highest growth pattern among the treatments, followed by 50 mg and 3 mg of urea treatments. For stem diameter, 0 mg of urea fertilizer produced the lowest stem diameter growth pattern, while 50 mg treatment gave the highest pattern. So the results are proven by studies from Menegheli 2017 and Merjad 2011, which states that the higher the rate of nitrogen treatments on plants, the higher the value of the mean stem diameter. Okay, next for leaf number, the lowest trend displayed by 0 mg of urea treatment, while the 50 mg give the highest trend among the treatments. It is supported with the ability of plants to develop leaf has risen due to the fulfillment of sufficient nitrogen. Next, I will continue to the relative growth rate. So, plant height, the relative height growth rate does not differ significantly between the treatments, with a mean of 0.314, and also there was no significant difference for relative leaf number growth rate, with value 0.067. So most of previous study have significance in plant height and leaf number parameters, but the value achieved in this study were not much different to be accepted. Okay, next for stem diameter growth rate. It was statistically significant difference among treatments for stem diameter growth rate. This result is supported by Menegheli 2017, which state the increasing of stem diameter shows that plants respond to the presence of nitrogen. Okay, and next I will continue to the plant biomass. Total dry biomass also represented a significant statistical analysis between all different urea treatments. 9 mg of urea treatment has a higher mean, which is 9.06 at above ground dry biomass among other treatments. The reasons to support this result is Solanum melongena is in flowering phase, so the nitrogen is absorbed a lot in the above ground part compared to the below ground part. And the last result is flowering stage. So observation throughout the study has shown the presence of buds and flowers in the 4th and 5th week, which is the last week of treatment. The presence of buds and flowers is only found in sample of treatment 2 and sample of treatment 3, which is the application of urea treatment 9 mg and 50 mg respectively. As the conclusions, 50 mg per pot, which is treatment 3 of urea treatment, is the best amount of nitrogen fertilizer, where it shows a positive effect on the growth of Solanum melongena plant for most of the parameters in this study. Alright, that's all from me. If you have any questions, you can reach me at my email and phone number. Thank you.